So, hi everyone, welcome to our first EBIC podcast. My name is Nicola Heaven. I'm the Science Communications and Knowledge Mobilisation Officer for EBIC. In case you don't know of us yet, the Environmental Biotechnology Innovation Centre, or EBIC, is a world-class interdisciplinary engineering biology research hub for the development of innovative environmental solutions. EBIC focuses on 4 tangible theme-oriented outcomes. Theme 1, biosensing. Theme 2, bioremediation. Theme 3, wastewater and waste management. And Theme 4, responsible research and innovation. Hopefully most of you know today is World Environment Day, which this year is calling for the collective action to tackle plastic pollution. So joining me here today are 2 of my colleagues whose work with biofilm engineering has a focus on using these tools to tackle environmental challenges such as plastic waste. So welcome to Professor Ronan McCarthy. Ronan is a reader in biomedical sciences at Brunel University London. Ronan, would you like to introduce yourself and your role within EBIC?
So my name is Ronan McCarthy, as you introduced. My role within EBIC is largely focused on Theme 2, environmental bioremediation, largely looking at using different genetic systems and genetic circuits to modify biofilm formation to target different environmental pollutants, but with a strong focus on plastic.
Perfect, thank you. Also joining us today is Lyuboslava Harkova. She's a research fellow and part of Professor McCarthy's lab at Brunel. Lyuboslava, would you like to introduce yourself and your work as well, please?
Yeah, thanks for that. I'm a research fellow at Ronan's group, and ever since my undergrads, I've been very interested in biofilms. I've spent my master's and 3.5 years of my PhD investigating biofilms in the context of human infections, so very much healthcare based. So now it's a bit of a challenge to shift my view on biofilms because my role in EBIC is to actually investigate biofilms and modulate biofilms or engineering them to improve degradation of various pollutants, including plastic.
Fantastic. Thank you very much. So, very excitingly, your work, Ronan, on bioremediation technologies was actually featured in Nature's 7 Technologies to Watch in 2025. And the article explores how your team are looking to use microorganisms to stem microplastic pollution. Can you tell us a little bit more about this work?
Absolutely. So a couple of years ago, my lab, I suppose, has a long history of looking at biofilm formation. And as Lyuboslava pointed out, much of that has been from a clinical context. So biofilms are a huge problem for people who get biofilm-associated infections. But within the environment, biofilms are ever present. It's the primary mode of growth that most bacteria like to undertake. And by doing this, they basically share resources between each other. They protect each other better. So we kind of had this idea that we have all of these enzymes that we know are degrading plastic, and we wanted to look at how we could optimize their activity. And there's some really exciting and excellent work out there where they're really focused on engineering the enzymes specifically. But we said instead we'd look at engineering the chassis, so the host for the enzyme, the host microorganism. And what we decided to do was actually try and augment biofilm formation in these strains. And the idea behind this was that if a bacteria is producing an enzyme and that enzyme is secreted into the liquid that's surrounding the bacteria, the enzyme then has to make its way to the plastic and then subsequently degrade the plastic as its target substrate. However, what we thought is if we drive biofilm formation in these bacteria, they'll attach to the plastic and then directly secrete the enzyme onto the surface of the plastic. And hopefully as well, within the biofilm matrix, you actually trap the enzyme so it can work there for longer. So that was the focus of the article that was highlighted in that Nature's feature article. And it was we subsequently proved that modulating biofilm can increase plastic degrading efficiency.
Fantastic. That's really interesting. So can you tell us a little bit more about the concept of biofilm engineering? I know, Lyuboslava, you've explained it to me before that it's almost like the house for bacteria. So if you could just explain that a little bit more for us as well, just the background there.
Yeah. So as Ronan just mentioned, biofilms are present in the environment. So naturally, bacteria they're kind of like us. They don't like to live on their own, so they don't like to live in a planktonic state. They like to group together and form their tiny little cities, as I like to call them. So biofilms are these cities or communities of microorganisms that live together, and microorganisms have this ability to produce a sticky matrix. It's almost like a cement that holds the different microorganisms together in these three-dimensional structures. So while in nature, biofilms act like houses for bacteria because they're protecting them from the environment, what we are trying to do in bioremediation and engineering biofilms is to kind of shift these houses into becoming more of a little factories that we can then specialize into different tasks, and we can just ask bacteria and microorganisms to do different things for us. For example, degrade plastic, and then they can go on and specialize this biofilm or this microfactory into doing this for us. So there are different components of the biofilm which we can modulate, but we are actually not modulating. To modulate the biofilm, we actually need to modulate the bacteria or the microorganisms that form the biofilms to begin with. So it's almost like we're just giving the bacteria or the other microorganisms specific tools, so then they can build the factories that are specialized for a specific purpose. In this case, it could be we can engineer parts of the biofilm that help microorganisms attach to the pollutant or sense the pollutant, so then they can form the biofilms only in the presence of this pollutant. We can give them specific enzymes for the degradation of this pollutant or for modifying the pollutant into another chemical or substance that we can then further use. So that's in a nutshell.
Yeah. That's perfect. That makes so much sense. So are there any recent sort of or key advancements that in the realm of sort of in bacteria, with plastic eating bacteria particularly, that have led to an increased interest for bioremediation, or any recent emerging trends, things like that, that are particularly promising?
Yeah, so I'm happy to take this one. So I think there was a real kind of explosion in the research surrounding this area about a decade ago, when there was a really interesting paper came out from Japan focused on a specific species of bacteria called Idenella sakiensis. And this was actually a bacteria that the research group identified by visiting a local plastic recycling plant and finding some plastic or taking some plastic samples and bringing those back to the lab. And when they initially did some exploration of what kind of microbial communities were living on the plastic, they identified certain species that were able to degrade the plastic that they were living on, specifically PET. So what was quite exciting about that work is they took it all the way to identifying the bacteria and then subsequently identifying the enzymes, so PETase, which has become a very important enzyme with respect to our understanding of PET degradation. So that was a real kind of landmark moment in plastic bioremediation by bacteria, and subsequently there's been countless additional enzymes identified, but also equally we've used other technologies like protein engineering and AI guided protein enhancement to optimize the activity of the enzymes that we do already have, because it's quite important to state that while we've identified more microorganisms that can degrade plastic, they're not particularly efficient at doing it, and I think that's really where the power of engineering biology and synthetic biology can be really used to enhance these microorganisms to really make them potential plastic bioremediation solutions.
Perfect. Yeah, that's something I was wondering about, actually, if there are specific challenges or actually also advantages as well when you're using biological agents for remediation.
Yeah, no, absolutely. So I think that there's two primary schools of thought around this. One is primarily focused on purifying the enzymes, optimizing the enzyme activity, and then potentially just using these purified enzymes for plastic biodegradation. And that is, I suppose, the fastest way that we can get develop solutions with this, largely because the purified enzymes themselves are relatively restriction and regulation free, and as such they can be used in I suppose bioreactor plants and these kind of type scenarios. The other school of thought is actually using the bacteria themselves, so that the enzyme, because producing enzymes can be a very expensive and costly process, whereas if the bacteria is growing and producing the enzyme itself, that may have more advantages. I suppose the limitation with that, though, is currently a lot of the bacteria that can degrade plastic don't do it very efficiently. So there's all these different approaches to try and enhance it and make it better. But the caveat with all of that then is if you are genetically modifying or engineering these strains, that then is associated with a wide range of different regulations with respect to their use. And I suppose their use and containment isn't as big an issue as the potential for, I suppose, environmental deployment. And that's where I think the real, I suppose, challenge is going to be is developing microbes in tandem with regulatory bodies that we can deploy, because a huge amount of the plastic waste is already out there. It's already causing these problems. And beyond us just going out collecting it, which is an okay strategy, but energy intensive and labor intensive, a real, I suppose, ideal type scenario would be we could deploy a microbe that could degrade it for us and could just specifically go to the areas of high plastic pollution and degrade those.
That's perfect.
I think
So,
if I can add something to that, sorry.
no, please.
I think a big concern would be then public perception, which we need to, as a whole, like the microbiology society, we need to put a lot more effort into improving the image of microbes, so to speak, because I've mentioned, modifying bacteria during outreach events to people from the general public, and their initial reaction is, oh my gosh, what are you creating in those labs? And then I proceed to explain that there's nothing dangerous. We work with bacteria that are non-pathogenic. We are actually just trying to just ask these little friends to help us. But the general public perception of modified microorganisms is currently not very good, and it's not really helping in terms of regulations or just scaling up and moving all this amazing research forward.
Yeah, so I suppose I know for us as well, it's important, isn't it, to consider public perception and the information that is out there as well, so that we can explain what we're doing and be quite open with the work as well. I think it's important that we're open, we're honest, we know. That there will be concerns when you mention, like you say, those sorts of words. But like you say, we can explain that they're our friends, bacteria. They can do very good things for us. Looking ahead, what are some of the most exciting future research directions emerging in the quest to combat plastic pollution, utilising biotechnology? Is there anything that you're particularly excited about or that you're very keen to work with?
I think one of the more exciting things is we're now beginning to see companies who can scale these solutions, which is the big challenge with all of this. And there are certain companies now that are capable of producing huge volumes of these enzymes that can then be used to tackle polyester waste or different plastic waste. So that's one of the more exciting areas. I think that scalability is definitely starting to come on the horizon. I think one of the very exciting things, and kind of harps back to our last point, is that within EBIC we bring together, let's say, expertise in bioremediation, but also expertise on, I suppose, containment and traceability of these organisms, which is a real something that has to go hand in hand with the development of microorganisms that will be capable of bioremediation. So we have, for example, digital barcodes or DNA barcodes that we're incorporating into some of these modified organisms so that we can trace exactly where they've gone. And also we're looking at, I suppose, creating kill switches within these organisms so that they can't go on beyond specific boundaries, or that they need specific nutrients such as some of these plastics to survive, and without those there, they very quickly reduce in the environment. Well, one of the, I suppose, if I was horizon scanning and thinking, okay, how are we going to really tackle this problem in the future? My personal view is we need to move away from these siloed centres of plastic degradation and bring it into the home. And I kind of have this vision that in 20, 30 years' time, everybody in their house is going to have something like a wheelie bin that we put all our plastic waste in, and we put a little pod full of enzymes and bacteria into the wheelie bin, and we come back a couple of days later and the plastic is degraded and it's either been consumed by the bacteria, or potentially we have some of the initial breakdown products of plastic that can be subsequently integrated into other waste streams or used to make high value products. That's the vision.
That's a good vision, to be fair. That sounds very good. I think a lot of people would be keen for that sort of thing, because obviously people are a lot more aware nowadays about pollution and the issues. Obviously everyone's doing their recycling at home anyway, they're separating everything hopefully, and kind of on board. So I can't see why people wouldn't go for that as well. I think that would be fantastic. So if oh sorry, no, you go.
No, so I do think it's worth flagging, because I didn't have a chance to do it yet, but we have this perception that recycling is doing a great job, but only about 10% of plastic waste is actually being recycled. And this is using the traditional waste streams, and beyond that, mechanical and chemical recycling of plastic is a very costly and expensive procedure. So if we consider that 90% of the plastic we're producing is either incinerated or going to landfill, it kind of puts a degree of urgency onto these kind of engineering biology-based solutions.
Yeah, absolutely. I think especially if people become aware of that fact, then they're going to be hopefully more interested to try and support the work as well, because obviously nobody wants to not be able to make the world a better place. Nobody wants to be leaving their rubbish everywhere and things like that. So that would be very interesting.
I think that would be awesome, because actually we can kind of divide bacteria into those that can completely consume the plastic and use this potentially for this home wheelie bin degradation of plastic, because that would be like complete degradation of the plastic, right? Bacteria would just literally eat it to create energy for them to grow and survive. But then on the other hand, in all the potential recycling, water or waste facilities, treatment facilities, we can have these engineered bacterial biofilms or bacteria or microorganisms in general that can upcycle. So rather than recycle, what they can do is break down for example, plastic into other components that can then be used to further produce other things. For example, what I'm thinking is, because there was a research published in the past few years that describes engineered bacterium that can degrade plastic, but then convert it into a compound that's needed for the production of nylon. And then nylon can then be used further to produce like a bunch of, in the textile industry, for example, for clothing, or it could be used to produce things like seat belts and all that jazz. So I think it would be interesting, because if we get to a point where we have bacteria eating plastic in our homes in the wheelie bins, it will kind of leave space for the waste treatment facilities to then be repurposed into upcycling pollutants and waste. And I think that would be a great thing to have in the future. It would be helpful, I think.
I like that. That sounds good. So if you were talking to somebody who's completely new to the idea of plastic bioremediation, what would be your key takeaway message regarding the role of bacteria, fungi, plants, everything like that in addressing plastic pollution? What do you really want people to know about their role?
I mean, I think the key take home is that many of the enzymes we need are already there. We just need to harness their potential. And I think that's where engineering biology and synthetic biology comes in. I think nature has developed these enzymes. We just need to harness them now.
Perfect. That sounds good. So before we finish, I do have some fun quick fire questions for you, not too serious. So If your research and work on biofilms can solve one everyday annoyance, could be anything like that, one annoyance overnight, what would you pick?
I suppose the the first answer that comes to mind is all the plastic waste we're generating. It's like you open your fridge and then everything is packaged individually in plastic wraps. Imagine just putting that in the bin and in 3 days it's gone. That would be fantastic.
Yeah. I like that. Okay, so do you have any kind of odd out there, anything like that, applications for biofilm research that you secretly hope would become reality?
I do.
Yeah.
So mine is actually using biofilms for clothing production.
Okay.
And this is actually an ongoing research, and people are looking into using biofilm, I should say, like produced by microorganisms that are not harmful to people, because otherwise it would be weird. But they're looking into harnessing the biofilms from these microorganisms, drying it out, and because of the components that bacteria produce in the matrix, in this sticky cement that holds them together, the qualities that the biofilms have could be suitable for clothing production in the future. We're very much not there yet, but I think it would be cool to just walk around wearing a t-shirt that's made up of biofilm.
I like that.
It sounds crazy at the moment,
no,
but
I
it
never
may
would
be a
have guessed
reality
it.
one day.
I like it. Why not?
Definitely buy one. I would definitely buy one.
I would, yeah. Sign us all up. So my last question. Okay, if you could give biofilms and bacteria a catchy slogan, we want to boost their public image, what would it be? You both can do this one if you like.
Big boob is
Mine,
good job.
yes, mine is micro factories tackling macro problems.
Oh, I love that. That is very good. See, we could put that on those t-shirts.
There you
Yes, yes.
There we go. We've got
So
a full
we have a
company
merch
now.
ready to go.
Excellent. That is perfect. Honestly, thank you both so much for your time today. It's been really, really interesting to learn more about your work and everything you're doing. And obviously, if anyone wants to find out more about EBIC, then do take a look at our website, which is ebicentre.co.uk, and do check out our socials as well. We do also have a membership scheme. It's completely free to sign up. Sign up on our website, and then we can keep you in the loop with all things environmental biotechnology. See you next time. Bye.
Thanks for having us.
Thank you.
Thank you.
Thank you.